Okay, hopefully it should record. Okay, so this is Unit 10, AHL Genetics. This shouldn't take too long, but I'll try my best to answer any questions that you guys have throughout the thing. You can keep messaging them in the stage chat, and I'll take a look at it after a while. Hold on. Session chal raha hai. Sorry. Okay, so first thing is independent assortment. I'm gonna assume a lot of you know the SL stuff, so I'm not gonna go through a lot of the like basic alleles and genes and stuff like that. I'm just jumping straight into the Unit 10 stuff. So the first thing you need to know is Mendel and his law of segregation and law of independent assortment. So law of segregation basically means that the two alleles of a particular gene separate into two haploid gametes during meiosis. So if you remember meiosis 1 and meiosis 2, after meiosis 2, during anaphase, the two alleles of a single gene split apart and make haploid gametes. Mendel discovered the law of independent assortment, which basically means that the parents that differed in two characteristics are controlled by two different genes. So because of the law of segregation, the two alleles separating, when parents are crossed, they will result in one specific allele in one specific gamete, which means that only that will be passed on, and that causes the independent assortment, depending on in humans, which sperm fertilizes the egg, or in plants, which pollen fertilizes the egg. That determines the independent assortment because that has only one specific allele. So when parents differed in two different characteristics controlled by two different genes, and those are crossed together, that is Mendel's dihybrid crosses. So for practice, if you guys remember what dihybrid crosses are, if you don't worry about it. I will explain it after this. But if you remember what a dihybrid cross Punnett square looks like, try to do this particular arrangement and put it in chat so I can check. If everyone wants to do that, I will leave this on screen. Can someone ping me when at least 2 people have responded so I can look at it, because I can't see chat while I'm sharing and it's a whole thing. I'll give you like 2 more minutes if anyone wants to try and attempt this. You can do it on a piece of paper and take a picture and put it in chat. If you don't want to, it's okay. It's completely fine. Does anybody want to attempt? That is a Punnett square, but and it's not a bad try, but this is a dihybrid cross, not a monohybrid cross. So it will differ by a little bit. But no one correct him or her, sorry. Don't correct them yet. And we will... Yes, I would like you to send it so that I can check if people understand and fix any issues. You can just put it in chat and I'll look at it. Ooh, someone did it. That's actually, I think, pretty accurate. I'm not looking at it too closely yet, but I think it should be. It looks to be pretty accurate. So, good job, Stanley. Does anyone else? Oh. Not a bad try, but it is. I did say heterozygous parents. And it's dihybrid, so there would be 4 and not just 2 here. But I'll show you in the next slide anyway, so it's okay. Ooh. Yeah, looks to be pretty accurate. I'll say it is. Just in most cases like this, I mean, I think you've done a pretty good job in the actual square, but at the top and on the side, try your best to differentiate between the two y's, because sometimes it can be more difficult than other times. I think you did a good job within the squares though, so it's okay. No, don't worry about your handwriting. I'm just commenting on that specific thing because that can cause you to lose marks for no reason, where you don't differentiate well enough between the two, and then that'll cause you, the examiner will just mark it as wrong. So, that's why. Yeah, no worries. Okay, I'll just move on for now. If anyone else wants to answer and give it a try, feel free to, and you can ping me, and I'll look at it. Okay, so this is what it's supposed to look like. I said 2 heterozygous parents, which meant, so here I gave a hint as well, but I don't know if anyone saw it. It was capital Y is yellow, small y is not yellow, capital R is rough leaves, small r is smooth or non-rough leaves basically. So if you do a dihybrid cross, the way it usually works is you have 4 alleles. So there will be 4 different combinations that can happen, because it might be a dominant Y and a dominant R together. It can be a small y and a recessive Y and a dominant R together, or it can be a dominant Y and recessive R and a recessive Y and a recessive R. There's 4 different combinations of pairs of alleles, and those pairs of alleles will then cross with another 4 pairs of alleles, which will result in this. So assuming that formation means rough, and the normal circle means smooth, and the green means a green plant, and the yellow means a yellow plant, this will give you a 9:3 to 3:1, which is the famous Mendelian ratio for phenotypes. So this is not the genotypic ratio, it's actually the phenotypic ratio. So if you should think back to unit 3 where you learn about genotypes and phenotypes, just make sure that you know what the question is asking for, because if it asks for the genotypic ratio, it won't be 9:3 to 3:1. It'll actually be a much longer ratio, because I mean they rarely ask for genotypic, but just in case they do, make sure that you know what you're talking about, because if you talk about phenotype when it's supposed to be genotypic ratio, it'll cause a lot of problems. So this leads to a 9:3 to 3:1 ratio, which is the general ratio that you can think of when you know the 2 heterozygous dihybrid cross, a dihybrid cross with 2 heterozygous parents are occurring. It's always going to be a 9:3 to 3:1 ratio, except when there's linkage or crossing over or recombinance, which is the same as crossing over, but we will talk about that after this. So the dihybrid cross ratio is the 9:3 to 3:1 ratio, which is often found when parents that are heterozygous for 2 genes are crossed together. So the ratio is the product of 2 3:1 ratios. So if you remember the monohybrid cross, which is the normal cross, where, for example, if a parent had rough leaves and the other parent had, if both the parents have heterozygous rough leaves, which means a capital R and a small r, and they cross, they will result in a 3-to-1 ratio of 3 rough leaf kids and 1 smooth leaf kid, basically. So so at the top here, that 3-to-1 ratio and that 3-to-1 ratio that emerges from their parents will result in a 9-to-3 to 3-to-1 ratio. In a dihybrid cross, they follow Mendel's law of independent assortment because they're unlinked. So now there's a difference between unlinked genes and linked genes, which I will talk about later. But does everyone understand up until here? Any questions? Anything I didn't explain well enough? Which, by the way, you can put questions in chat even when I'm not looking at it, and I'll look at it right after. Someone's typing, so I'll just wait until they're done. And then, in what case would a heterozygous cross not fall in the... In what case would even if it's not unlinked? Even if it's not I don't think there should be any difference if it's not unlinked. Unless I'm horribly wrong and someone wants to correct me. But if it's an unlinked heterozygous cross, it should usually be 9-to-3 to 3-to-1. Let me think about this. Hold on. If it's... Yeah, I think it should be 9-to-3 to 3-to-1. If I'm wrong, I will correct myself later on. I'll think about this after the session and I'll ping you in chat. Yeah, exactly. Okay, yeah. Obviously, if there's crossing over or recombination, even when it's unlinked, it can cause differences in the Mendelian ratio. That makes sense. Sorry, that's my bad. Because crossing over can occur even if it's not linked genes. It can just happen anyway, if that makes sense. Anyway, now we come into linked and unlinked genes. So genes that assort independently, so that follow Mendelian's law of independent assortment, are unlinked genes. Genes will assort independently if they're located on different chromosomes. So as you can see here down below here, the not linked, one of the gene is on this one and one of the gene is on this chromosome. They're on 2 different chromosomes, or in this case chromatids, but let's talk about chromosomes. In 2 different chromosomes, which means that they're unlinked for obvious reasons, since they're on 2 different chromosomes. Gene linkage is caused by pairs of genes being located on the same type of chromosome. So for example, if we take the example of this cross, if we assume that Y and R and small y and dominant Y and dominant R and small recessive Y and recessive R are linked, then they would be basically gene 1 and gene 2 would both be here. So we would assume that this would be capital Y and this would be capital R, or lowercase y and lowercase r. The scientific name for the location of a gene on a chromosome is a locus, which you should know. So linked gene have loci on the same exact chromosome. So in the non-linked, their loci are on 2 different chromosomes, on 2 separate chromosomes. But for linked ones, they're on the same exact chromosome. And usually they're really close together because that's how it attributes to crossing over and issues with that. You would still consider it non-linked if they were on the same chromosome but very far away, but we don't have to learn about that. So just think of it this way: if it's on the same chromosome, it's linked. If it's not on the same chromosome, it's not linked. So the dihybrid pattern the dihybrid cross will change depending on whether or not it's linked or unlinked. So at the top here is unlinked genes, right? Which means that it can be any arrangement of the 2 alleles, and it doesn't have to be one particular arrangement. So then because of how meiosis and then fertilization occurs, so these 2 sister chromatids would separate, and then these 2 chromosomes might separate or might be in the same, or it can all change depending on what the arrangement is. That assortment, which is the Mendelian's cross, will result in the 9 to 3 to 3 to 1 ratio. But for linked genes, since they're linked together, they have to be combined together. So in this case, the uppercase A and uppercase B are together, but it can also be uppercase A and lowercase b, or lowercase a and uppercase b, or both lowercase a and b. In this situation, since A and B are on the same chromosome, and that chromosome has to go to one of the children, it means that the child will definitely have the A and B on the same chromosome, and will definitely have only the pair of capital A and capital B, which means it results in this. Does everyone understand this? Does independent assortment just mean that it's random which allele from the parent? Yes, that is what it means. So if we go back here, one child could receive one child could receive capital Y, capital R, but one child could receive small y, small r. And that just, it depends on which sperm fertilizes which egg, or which allele goes to which child. Yeah, no worries. Okay, I'll go back here. Okay, so do we need to know Mendel's laws by heart? I don't know if it's ever been asked before. I haven't seen it before, but you should know. I mean, you should know what it generally means. I don't think you need to know exactly what the words are. This I have copied from the textbook, but you don't have to know exactly what it is. You just need to know what it means and what it entails for genetic variation in general. Because chapter 10 is more about genetics and how it contributes to evolution. So that means that in the middle there's the process of variation, right? So you need to know the Mendel's laws and how they attribute to variation, but not not them by heart. Are the dominant alleles supposed to be on the same chromatid for crossing over? Usually, but they don't have to be. I don't think. I think it could very well be that the capital A and the small b are together, but once they're linked, that means that they will stay together. This mostly we say that the dominants are together and the recessives are together, but I don't know if it's necessary that they have to be together. It just means that the alleles have to be on the same chromatid in general. I don't think they have to be both dominant. Am I understanding your question correctly? Because I don't know if I'm understanding it correctly. But if you mean that they have to be both dominant, then no. But they have to be on the same chromatid, otherwise they wouldn't cross over, which I will explain now. Which I will explain crossing over. Okay, so going back to meiosis. In meiosis, obviously there's interphase right before the meiosis or mitotic phase, because it can happen in both mitosis and meiosis. Interphase happens in general, but specifically before meiosis, it causes DNA replication, right? So each chromosome will then have two chromatids and form sister chromatid pair. That happens in the S phase. Now, about recombination and crossing over. So, new combinations of the alleles of linked genes can only be produced if DNA is swapped between chromatids. This is called recombination, and it happens by crossing over. So, I'll explain what crossing over is, but basically, if the individuals that have a different combination of characters from parents are recombinants. So, for example, if your parent has a capital A, capital B, or a dominant homozygous A and B, and you have a heterozygous dominant A and recessive B, that means that there has been some crossing over or recombination happening, and you are therefore considered a recombinant. And by you, I mean your gene. So this on the right is the process of gene recombination. So for example, you can see this set of sister chromatids has both capital A, capital B on the same side, Here it's small a, small b on the same side, but due to crossing over, it results in these four completely different sister chromatids. Without crossing over, it would result in two pairs of sister chromatids, because the sister chromatids would be identical. But due to crossing over, and due to how meiosis works, it results in recombinance, which means that there will be four different alleles. This leads to more variation. So crossing over and recombination is a method of variation, basically, which is why when you talk about natural selection and variation in unit 5 as well, and in this, you talk about how meiosis plays a part in genetic variation, because it causes a high percentage is crossing over. So crossing over is the exchange of DNA material between non-sister homologous chromatids. I know it's a mouthful, but basically, because there are homologous chromosomes in in gametes before meiosis occurs. There's homologous sister chromatids, which there's a homologous pair, and then there's sister chromatid pairs. So this red and the other red are considered the sister chromatid pair, but the red and the blue as whole units are homologous chromosomes. So first of all, in synapses, the homologous chromosomes pair up together in prophase 1 of meiosis. When they pair up, they are wound really tightly, which means that they basically are next to each other. Like, they have this space that you see between them might not exist. They're that tightly wound together. And then the DNA molecule of both of the chromatids is cut at the same point. So if you look here at this point, both of them in both of the sister chromatids of opposing homologous chromosomes will be cut, and they will basically exchange and swap around. So in this situation, the blue will go to the red will go to the blue, and then that will result in this combination, and which will separate into this later on. But basically, the point at which this exchange occurs, the point at which it's cut and the point at which it exchanges, is called the chiasma. I don't know how you pronounce it, but as long as you know how to spell it, it doesn't matter. You need to know how to spell it. That's the site at which the non-sister chromatids are still adhered after crossing over at the end of prophase, but they separate, obviously, right, I believe, at metaphase. So when they line up in the middle and then get pulled apart. I'm gonna call it a break. If you guys have any specific questions, it doesn't have to be related to genetics. Any questions, you can ask me. And this is basically a break. Everyone go get water. If you have questions, you can ask me. But I will continue in. I'm gonna say 5 minutes, but if there's not a lot of questions, I'll just continue on right after. What is a polyploid zygote? Which cell is a polyploid zygote produced by fusion of one haploid and one diploid gamete? I will explain what polyploid stuff is later on. Is chiasma only visible during prophase? I believe so. I can cross check just in case it's not. At what point does the chiasma split? During anaphase 1. So chiasma is visible at all points up until anaphase, at which point when it's pulled apart the chiasma will split. Am I planning on going over any other topics? I might, depending on how much time I have and how what people are most interested in. If you have specific topics you want me to go over, you can just either ping me specifically in chat, in the N24 chat, or you can DM me, I don't mind. But yeah, and next weekend, right before the exams begin, I'll probably do like a Q&A question with maybe one Lipid or someone else from the who's the session leader as well, and you guys can ask any general questions, and we'll try our best to answer. When a question says test breeding or something along those lines, does this mean that there's one dominant homozygote parent and one recessive homozygote? Usually, I think so. Or no. Yes. I think test breeding does mean that, but let me check. I don't want to spread false information if I'm wrong. Hold on. Just give me a second. I have to stop streaming. Let me check. Where is my... Oh, no, not you. Okay. Test cross using a plant that is homozygous recessive and... Okay, no. A test cross means that when you have one specific, the genotype of one of them, or you're trying to find the genotype of one of them, you cross them with a homozygous recessive, and then figure out based on that. So it's not, when it says test breeding, it doesn't mean that one has to be dominant homozygous, but it does mean that one of them has to be recessive homozygous. So the first one can be anything, but the second plant or animal or whatever is used for the test cross has to be recessive homozygous. That just is what it is. It gives you the best results, I think. Because when you cross it with a recessive homozygous, you can tell whether there was any because okay, so for example, if you have a homozygous dominant parent and a homozygous recessive parent, they're going to give you all heterozygous children. Yes? And if you have a heterozygous parent and one recessive homozygous parent, that's going to give you a different ratio. I don't remember the exact ratio, but basically it'll because you'll get a specific ratio of heterozygous kids and a specific ratio of homozygous recessive kids. So it's the best outcome, because if you cross with a dominant homozygous parent, then you're just gonna end up masking all the traits of the first one, of the one you're trying to figure out the traits for. If you just mask everything, then there's no way for you to trace it back and try to figure out who is what are their genetic makeups. But if you cross them with a homozygous recessive, then nothing's masking it, so then you can tell whether or not there was any heterozygous or homozygous. So dominant I'll read this in chat, but dominant homozygous means that it's both, zygous, I cannot spell. Homozygous parent means that it's capital A. Recessive means that it's small a, and Heterozygous means capital A, small a. So if you cross either the heterozygous or the dominant with recessive, you'll get two different results. But if you cross any of those with another dominant, you'll get the same exact result, because it'll just the dominant will just mask over everything else. Does that make sense? Yeah, no worries. Feel free to ask me more questions as we go along. But I'll assume most people are back from break if they left. I don't know. But yeah, so I'll continue. If you have any more questions, keep putting them in chat and I will answer them as I go along. Polygenic inheritance. So everything after this is more about evolution and distribution of genetics and distribution variation, essentially. I'm not going over the chi-square test just because I personally don't like it very much, but if you want me to go over it, let me know and I'll just pull up one of the pages from my study guide and try my best to answer stuff. But I'm not going over chi-square right now. So polygenic inheritance is when the combined effects of two or more genes leads to a continuous variation. So not going into this particular example, but if you want to think about continuous variation, think of, for example, heights of people. So the heights of a specific population can be continuous and there's an overlap between the frequency, even if they don't have the same exact one specific gene. And that's because there's multiple genes that contribute to that one particular characteristic, and there's also environmental factors which basically both together affect how the characteristic occurs in humans or plants or anything else. So this particular example is for wheat grains, where wheat grains can vary in color from white to red, as you can see on this graph here, from white to red, and this is the frequency. So if you guys do math, then you'll know what normal distribution is. I mean, everyone does math, but if you guys remember from math, the normal distribution just means it's going to be an equal spread throughout, essentially. So in wheat grains, there's three genes that control the color, but each of those three genes has two alleles, one dominant, one recessive, for obvious reasons. So when there's three different genes that are affecting just one characteristic, that means that there can be anywhere from 0 to 6 alleles for pigment production. So how many alleles one particular plant or wheat grain has can change how pigmented it is. So for obvious reasons, I say obvious reasons, if you have any questions you can let me know, but for obvious reasons, the most is the middle, because most of the plants, I presume, would have 4 alleles, which would then mean that they have an equal distribution of white and red. I'm assuming, I could be really wrong here, but basically you just have to know that there's polygenic inheritance, and that's also affected by environmental factors. So polygenic inheritance is caused by multiple genes that lead to one specific characteristic, and environmental factors that also affect that characteristic and therefore lead to a normal distribution of inheritance. Is there any continuous variation is always normally distributed? Yes and no. I take math HL and I'm struggling with this. That tells you the status. I don't think so, but here just think of it as continuous distribution, which is like this. Okay. Gene pools. So a gene pool consists of all the genes and their different alleles in an interbreeding population. So the gene pool can have, so if we go by let me go back. If we go by this, for example, so dominant A, dominant B, recessive A, dominant B, No, basically think of it like this: dominant A, dominant B, recessive A, and recessive B are the four alleles, and all of them are part of the gene pool in an interbreeding population. So, any new individual that is produced by sexual reproduction inherits genes from its two parents. We know that. That's how inheritance works. Assuming that there is random mating, so in a wide population of animals or plants, you don't know who is mating with who, then any two individuals in an interbreeding population could be the parents, because you don't know, obviously you're this is not like human relationships, so it doesn't mean that they have to mate with the same person. So any two individuals in an interbreeding population could be the parents, which means that an individual that is newborn can have any of the genes in the gene pool. So if you want me to be more specific, for example, if in the entire gene pool there's capital A, capital B, small a, lowercase b, the four alleles, and they're randomly assorted in a population, it's possible that the new found individual could have any combination of A B's, in terms of, basically, if you think about the dihybrid cross, the four alleles combinations that are possible, the offspring could have any combination of those four, and you wouldn't be able to trace it back to the parents unless you do a full genetic analysis, because the gene pool consists of all A's and B's. So you don't know whether or not one specific child belongs to one specific parent unless you do full genetic analysis. So in a typical interbreeding population, some alleles will be more common than others. And evolution always involves a change over time in allele frequency in a population's gene pool. Does this make sense, or is there any questions? Because I feel like I wasn't as clear, but if everyone still understands, then I'll keep going. Okay, I'm gonna take that one answer and move on. If you have any questions, obviously, put them in chat. Okay, so there's 3 different types of natural selection: directional, stabilizing, and disruptive. So directional natural selection is when if you want to think about it in terms of direction, since the word says directional, one direction in the range of the variation is selected for and the other extreme is selected against. So for example, if you think of the shaded area here in terms of think of the shaded area as neglected or selected against, then that means that this specific extreme is favored. So all of the white portion is accepted, but then the shaded portion is selected against, which means that you'll just naturally have more of this population and less of this population. And an example of this was an example of this was a specific bird favors those who mate early rather than mate late. So for example, if you think about this, the shaded area is mating late and this means mating early, and these are just favored naturally because of early mating and being able to mate only once in each season or something like that. So early maters are favored, and anyone who mates late are basically selected against, since their alleles will not be passed on. These people have mated already. These are not mating, which means that these alleles will not be passed on. These are. Stabilizing is when both of the extremes are neglected and only the intermediate is selected. So in this situation, only one side was neglected. Here, both of these are selected against, and only the middle part, which is around the normal, will be selected for, which means that the allele frequency will be much narrower because it will be specific to only the most frequent allele, if you think about it that way. Disruptive is the complete opposite. So if you see stabilizing like this, disruptive is the complete opposite. The center is completely selected against, and the extremes are selected for. If you think about the nature of the curves, if you the and consider the heights of the curves, the height means the frequency. So the least frequent here is rejected, and this least frequent is still maintained, which results in an overall average curve. In this, both the extremes are neglected, which means the one that is the most frequent will just keep being produced, which reduces the variation. And then the worst is disruptive, where you remove almost all variation and leave only the two extremes, which are already very few in frequency, and if they only those are favored, it will just the gene pool will just decrease even more. Speciation, this is the last thing that we do. The formation of new species is called speciation. So this is when a pre-existing species, so this is part of unit 5 as well. I haven't gone through that, but if you remember cladistics and how one particular species can divide into multiple species, that's speciation. So new species are formed when pre-existing species split. This usually involves one population not interbreeding with any other populations, which is also known as reproductive isolation. So if one population of a species and another population of a species, there's one that interbreeds with the rest of the populations and one that doesn't, for many reasons that I will go through, does not interbreed with the rest of the populations, they'll just interbreed within themselves, which will result in narrowing the gene pool, since they have only themselves to interbreed within and not the entire gene pool of the species, just their population. So they natural selection will act differently on this population, and it will diverge gradually from other populations, and then eventually this isolated population can't interbreed with the rest, since their gene pool is completely different to the completely varying gene pool of the entire species, so it becomes a whole new species. Does that make sense before I go into reproductive? What is natural selection? Part of unit 5. Yes, what Jiffy said. Okay, there's 3 different types of reproductive isolation that you need to know: temporal, behavioral, and geographical. So temporal is when populations of species breed at different times. So for example, in one area, if one specific population prefers to breed in another year or in a earlier part of the year, while the rest of the species breeds at the end of the year, they will only be able to breed with themselves, which will result in isolation in and of itself. While the other species, since they interbreed at towards the end of the year, they will interbreed with each other, which will largen the gene pool, which will result in a continuous variation. Behavioral can be when populations of a species have behavior that prevents interbreeding. So this is specifically for birds. If mating calls that are used to attract females are different, just within a species, they can be different in different populations, and different females will favor different calls. The species will split eventually, since one type of female or one population of female will prefer one type of mating call and the other will prefer the other type of mating call. They will split into 2 different reproductively isolated populations with distinguishable mating calls, since one will only be attracted to one. Geographical is when populations of a species live in different areas. So it can just very well be that due to some migration, this is underneath it says from island to island, but even if you think just within an island, if an island has a mountain range and the same species population has been split to either side of the mountain range, they're not going to be able to interbreed for obvious reasons. So then that lack of interbreeding between them splits their gene pools, and then therefore it splits them into different species. Does reproductive isolation and speciation make sense? And then the last one is speciation by polyploidy, which is the question that Robert asked before. And then we're done, basically. So polyploidy is when an error leads to an individual having more than 2 sets of chromosomes. So for most people, we all have 2 sets of chromosomes, 2 pairs of chromosomes. So we have a diploid number of 46 because we have 23 pairs of chromosomes. In some plant groups, however, there is a trend for species to have chromosome numbers that are multiples of one basic number. So it's possible that instead of having a diploid number, they can have a tetraploid number because due to some errors, they've replicated twice. So in a species with a diploid number of 16, which, by the way, this is an example of allium, I believe. So if they have a diploid number of 16 and there's another individual that is created from that same species, perhaps, with an individual of 32 chromosomes, which is basically 2 times of the diploid number, it results in a tetraploid number. But then if the tetraploid and the diploid cross, because one has 32 and one has 16, when they cross, they will result in triploids, which will basically be 3 sets instead of 2 or 4. Because they're 3 sets, they can't fertilize another because they can't split into equal haploid there's no version of haploid. Even if a tetraploid crosses with another tetraploid, they'll just divide and make diploid gametes. But if a triploid individual tries to make gametes, they won't be able to make equally halves of, because you can't have 1.5 chromosomes. It has to be 1 or 2. So they basically will be infertile. So because of this, a tetraploid can only participate in sexual reproduction with another tetraploid, and a diploid can only be with another diploid, and therefore when polyploidy happens, it causes instant speciation. So speciation there's 2 different types if you think about it. There's gradual and there's instant, and this would be instant speciation. There's gradual speciation, which is the normal type of like reproductive isolation, because it takes a while, and the entire process of isolation and breeding and interbreeding will take some time, and the gene pool will slowly lessen. But in polyploidy, as soon as polyploidy has occurred, there is no way for them to interbreed at all, which means that many plant species will separate, and there have been many plant species that have been produced due to polyploidy. And that's it. I'm basically done. So if you guys have any questions, I'm willing to answer them. If